Sciences at Harvard. There we go. Got it. Where she worked with Jeffrey Parvin and did a lot of nice work on BRCA1, which is, as many people know, a DNA damage repair, a gene that plays a very prominent role in breast cancer. We might even hear a little bit about that one today. But then she went on, after getting her PhD, to do a postdoc in Stan Field's lab. And as many people know, Stan is really has been a pioneer of clever approaches, including one called deep mutational scanning. Lea was an early pioneer working in developing and using that technology that really has fostered a lot of her ongoing research today that we may hear about. So Lea is, as I mentioned, she is an assistant professor. She has her own lab, but she's also the co-director of the, let me see, the laboratory, the Advanced Technology Laboratory at the Brontham Beatty Institute for Precision Medicine in Seattle. She's the co-PI of the Seattle flu study. And as you can imagine, you might already know, diverted quite a bit of her efforts. And during the COVID pandemic, Seattle was really one of the hubs of COVID epidemiology and genomic research, and Lea's group participated in that. She has leadership roles in a number of different consortia, working groups. Many of those are specializing in trying to predict and annotate and catalog human genetic variants that have important phenotypes. And Lea is also the co-PI on one of those, in one of those projects that we have a UW team participating in. And this is called the, within the NHGRI at NIH, the consortium called IGVF, studying the impact of genomic variation on function. And so it's really great, fun to have Lea here visiting in person today, and I'm really excited to hear more about her work. So with that, I'll turn over the stage.
Thanks, Audrey, for that really nice introduction. It's always nice to be at the other UW. As I was telling the students earlier, sometimes I Google UW something and I'm like, this webpage looks red instead of purple. What's happening here? And I find all sorts of websites for y'all. Okay, so I'm going to try to use the Zoom pointer so Zoom can see if that's okay. All right, and as Audrey told you, I'm going to be telling you about clinical variant interpretation and the challenges they face in trying to understand what they're reading, what the DNA sequence changes actually mean for human health, right? So sequencing has gotten pretty inexpensive. It's now used more and more in the clinic, and whether it's whole exomes for trying to discover rare disease genes, or it's gene testing panels or genetic testing panels for things like hereditary breast cancer or cardiovascular disease. A lot of variants are found, and the thing that can happen after a genetic test is you can get one of these 5 answers, right? You can get a variant that is benign, which means it has a less than 5 to 10% chance of being disease causing. You can get a variant that is likely pathogenic, meaning that we are more than 90% sure, and there's actually some Bayesian math under there, 90% sure that that variant is disease causing. And there's this vast category in the middle between 10 and 90% certainty that a variant, we just don't know. And those are the variants of uncertain significance. And it turns out that a lot of the variants that we find are variants of uncertain significance. So this is the contents of the clinical genetics database ClinVar at the moment for missense variants, right? So these are variants in protein coding genes. So these should be much easier than non-coding variants to understand, but still harder than things like nonsense or splice disrupting. And there's 550,000 of them, and 80% or a little bit more are actually variants of uncertain significance, right? And this problem is only getting worse. So if we look at the contents of ClinVar from when it started in about 2014 to 2022, my graduate student remakes this slide for me very kindly every year. You can see that the variants of uncertain significance are rising exponentially, whereas the variants that are in these more definitive categories of pathogenic or benign are kind of rising linearly, right? And I think we're in this problem because the more and more we sequence, the more rare variants we find, right? So variants that don't have enough evidence to push them into these definitive categories. And I think this is the opposite of what we thought we would do, right? We thought we would sequence more, we'd be able to link more variants to disease. But as it turns out, most variants are pretty rare. So if you think about the size of the human population, and then you multiply that by the de novo mutation rate in humans, you get about 50 people walking around with any possible single nucleotide variant that is compatible with life, walking around on the planet, right? And by sequencing more and more people, we're finding these folks with these rare variants that we can't do anything about. And I think that's what this is showing us here, this exponential rise in variants of uncertain significance. And this has an impact on our ability to practice all of this precision medicine that we said we were going to do when we sequenced the human genome 20 years ago now, right? And so this is an a Kaplan-Meier curve of people who either got surgery when they found that they had a pathogenic variant in BRCA1 or 2, or didn't. And you can see that that surgery is life-saving for these women, right? So hereditary breast and ovarian cancer or pathogenic variants in BRCA1 and 2 are highly penetrant for breast cancer. So this is like the OG of precision medicine, right? But if you look at the contents of ClinVar for BRCA1, 85% of the missense variants are variants of uncertain significance, right? So this is a gene we actually really know something about. We know what it does. We've sequenced more than 1 million women and families for this gene, and yet this is what ClinVar looks like for BRCA1, right? And these VUS are a missed opportunity to either give these people care or tell them they have nothing to worry about. Okay. Yes. All right. So how are we going to find evidence to move these variants into either one of these definitive categories? Well, pedigrees would be amazing, right? If we had big families and we can follow the variant through the family and whether or not these people get breast or ovarian cancer, that would be awesome. But this isn't going to happen, right? Families just aren't this big anymore. Cascade testing is not something that U.S. healthcare is going to pay for ever, as far as I can tell right now. And so these sorts of pedigrees just aren't happening. And then the other thing you can do is you can sequence a lot of people and you can say, oh, well, this variant occurs at a super high frequency. It cannot be a highly penetrant hereditary breast and ovarian cancer variant, right? So that has to be likely benign. But again, there's certain populations who are excluded, who have been historically excluded from these big genetic studies. So we know nothing about variants in those populations. And, even again, most of these variants are very rare, so you're never going to aggregate this kind of information. Another piece of evidence you can get is through functional data, right? You can actually make that protein variant, or if it were a promoter variant or an enhancer variant or something like that, you can make that variant, you can test its function in the lab. And the way that it has been done historically is that these variants kind of pile up in genetic databases or through genetic studies. And then eventually somebody 10 years later goes and makes that variant in a test tube or a mouse and says, oh, well, we've either broken this protein or we've broken this promoter or we haven't. And that these one-at-a-time studies just aren't going to keep up with the rate at which we're discovering these variants. But that same high throughput sequencing that has enabled all of this clinical genetic testing has also enabled a type of experiment where we can test hundreds to thousands of these variants all at the same time. And those are multiplex assays of variant effect. And by multiplex, I mean all of the variants are together in a pool, so you're testing them all simultaneously. So if we imagine that each one of these dots is a different variant in a cell, you can subject all of those variants to some functional assay. And in this I'm using growth. So maybe this blue variant is good for the cell, and you see it become more abundant in the population. Maybe this red variant is not, and it becomes depleted. But the functional assay can be anything, right? Proteins come in all sorts of different flavors and functions. You can hook up anything as long as the genotype is connected to the phenotype, because we're going to use DNA sequencing to both identify and count the variants in each of those populations, so that we can tell what is happening during the selection process, right? So this could be FACS. You could sort things into FACS bins using reporter genes. You could look at RNA output of promoters, that sort of thing. Okay, so again, we're just counting these variants, and a functional score, so I'm going to talk about functional scores a lot, is essentially just the ratio of the variance abundance in the population after selection to its abundance before selection, right? So in this case, we're going to get for the red variant that became depleted, you're going to get a negative or less, a ratio of less than 1, and that would be negative when you turn it into log 2. Okay. So we have these multiplex assays of variant effects, and our goal is to end variants of uncertain significance as we know them, right? Really find, enough evidence to be able to move these variants into definitive categories so that we can enable precision medicine. So how do we get there? So I want to talk to you first about a success story using MAVES. And next, after that, I'll talk a little bit about how we're trying to scale them, new technologies to unlock new phenotypes, community building efforts to democratize and propel more labs to be doing these sorts of things, and also how to get that data into the clinic, right? Apparently, doctors do not want to look up supplementary table 2 in your paper, right? So I'm going to talk about some of our efforts here in this effort. So first, I'm going to talk about that success story for MAVES. And what I'm showing you here is the haplotype or the karyotype of a haploid cell line called HAP1. And so growth assays had been used in yeast and bacteria really successfully for deep mutational scans. Yeast are, the awesome power of yeast genetics really let you get in there and make huge libraries of variants. But human cells express human proteins, which is where we are, where the proteins we're trying to study. And although we're never going to be able to get to the scale that we can get to in yeast, this haploid cell line is really nice to be able to. It's almost like a yeastification of human cells. And you can actually see the Philadelphia chromosome here. It was a leukemia at some point. And so it turns out that a lot of genes that I care about happen to be essential in this cell line. So if we look at the essentiality from a study from the Brummelkamp lab from 2015, you can see that all of these variants here at the end are essential. And then all of the ones with red tick marks are in the double-strand break repair pathway. And all the ones that are labeled are genes that are on hereditary breast and ovarian cancer testing panels, right? So all of these, so BRCA1 and its buddies over here are all essential in this cell line. And so we figured if we could edit these variants, edit variants in those genes into this cell line, we would be able to use whether or not this cell lived or died to be able to tell if those variants were damaging to the protein function. And the way we're going to get those variants into BRCA1 was a method called saturation genome editing, which was developed by a graduate student in Jay Shenduri's lab during his rotation. So what we do is we take a guide RNA, a guide and CRISPR-Cas9 targeted to an exon in BRCA1. In this case, this is exon 18. This is the exon structure. These exons at the 3' end encode one of the important functional domains of BRCA1. These 3 exons here at the other end encode the other 3 exons. The rest of this protein is kind of a wasteland. It's huge, but it doesn't do much. So we make a double-strand break. And then we also give these cells a plasmid library of repair templates. And what these repair templates are, they're big homology arms, the sequence of the targeted exon, and then each one has a single nucleotide variant that we cloned in using programmed arrayed DNA. And as the cell fixes this double-strand break, these variants get into the genome, right? So these variants are now in the genome. What's really nice is because we're looking in the genome, we're not expressing cDNAs the way that a lot of these mutational scans have been done classically. We can look at variants that affect splicing and that are outside of the coding region, which is really nice. Okay. So now that we've got edited cells, we sequence the cells at day 5, and then we just wait for about a week and sequence the cells again at day 11 to see which variants have become depleted from the population. And I'm bringing this up now because it's going to become important later. Each one of these editing experiments requires a guide and a SNP and a repair template library for every about 120 bases that we target. So this is fairly labor intensive, right? So these 13 exons that we targeted will give us data for 4,000 possible single nucleotide variants, which is awesome, but it also is a decent amount of work. You have to have a guide and a library for each one of these targeted exons. So, but in the end, we did generate functional scores for 4,000 variants of BRCA1. Here at log 2 of 0 is essentially no change. Log 2 of -2 is where in red the nonsense codons are piling up. So that's good. We've depleted the nonsense codons. That's a great sanity check. Those are nicely separated from synonymous codons or synonymous changes, which are in light blue, although about 1% of those were damaging enough to splicing to become depleted from the population. And you can see that the missense variants are a nice bimodal distribution around those two populations, and also variants that once you get kind of closer to where the canonical splice sites are, those are also pretty bimodal as well. But about 80% of the variants had no functional loss, and 20% were as bad as stop codons. Okay. And that is the data. That was the data in aggregate, but this is what we call a sequence function map, and it gives you a highly detailed view. This is exon 2, and I'm going to blow up the splice donor here. And so the sequence function map, this is every nucleotide position in exon 2. This is if they're green, they're missense variants. If they're yellow, those are those canonical splice sites. So those two nucleotides on either side of the exon that are like absolutely essential for splicing. The reference base is the written base, and then each of the squares represents a single nucleotide change. So this would be A to C here. If it's filled in, that is a variant we classify as non-functional or functionally abnormal, I should say, because it overlaps that nonsense variant distribution. And we have a little bit of mechanistic information here. So these guys with these red bars in them, we also sequence the RNA after editing, so we can tell if the RNA has become depleted. And so these variants, not only were they not compatible with life, but these guys with the red tick marks, it was because the RNA was depleted. And that makes sense here. If you mutate a G that's at the end of an exon, that usually causes bad things to happen to splicing. But these sequence function maps are a lot of information, right? This is almost every possible single nucleotide variant in exon 2, provides a lot of information for somebody who might want to be looking up this information to understand what's going on with variants they find in clinical testing. So I've told you we use this haploid cell line. This haploid cell line happened to be a leukemia. It has nothing to do with hereditary breast and ovarian cancer. So we wanted to be able to tell, like, are we measuring the same thing in a breast and ovarian cancer risk protein in this cell line as we would, is the same function that's causing these cells to die or survive the same function that BRCA1 needs to be a tumor suppressor for hereditary breast and ovarian cancer. And so what we did was we took our functional scores and we looked up all of the variants that were in the clinical genetics database, ClinVar. And we asked, how those performed in the assay. And you can see all of the benigns and likely benigns separate pretty nicely from the likely pathogenic and pathogenic variants. And again, this non-functional, or what we would now call functionally abnormal, is the variance that overlaps the distribution of the nonsense variants, and these functional ones, which we now call functionally normal, would overlap the synonymous variants, right? So this gave a really nice separation. When we looked some of these variants up, they were clearly ClinVar mistakes, actually. So it's a really nice separation of both of those variant classes for very high specificity and sensitivity of the assay. So I said that we've kind of built these things to be able to reinterpret variants of uncertain significance. Like how do we do that? So I have a grad student, Sean Fair. He's co-advised by myself and Doug Fowler, and he is a clinical genetic counselor. And he worked at Brigham and Women's Hospital at Harvard and the adult genetics clinic. And he was tired of giving VUS results back to his patients. And so he actually came back to graduate school and has done his PhD in my lab. And this is his work here, where he took all of this functional data from BRCA1 and from other genes that I'm not going to talk about. And he tried to figure out, well, just how useful are they in clinical variant interpretation frameworks? Okay, so the first thing he did was collect and curate functional data for clinical integration. And this is kind of boring but important stuff, right? So like making sure that the variants are named correctly so that when you go to look up variants from a genetic testing company, they have the same name. But all of a lot of biology or computational biology kind of lays on that foundation of the foundational work of making sure all of the annotations are correct. The second thing he did was calculated the strength of evidence that could be applied to the data from that assay, right? So ClinGen, which is the NHGRI's clinical genetics resource, says data can be supporting, moderate, or strong, and that is how much weight it carries in the clinical variant interpretation framework. So supporting doesn't carry as much weight as strong. And that can be, so for functionally abnormal, we can apply that toward the pathogenic direction, and functionally normal, we can apply any one of these codes toward the benign direction. Okay. And then he's going to take that strength of evidence and add it to other evidence for each one of those variants and reinterpret those variant interpretations according to the American College of Medical Genetics. Okay, so I showed you this plot for BRCA1. ClinGen says there's like a kind of a modified odds ratio called the odds of pathogenicity. It's a Bayesian framework that quantifies how well the assay is able to discriminate between known pathogenic and known benign variants. And the more the assay gets right, the higher the strength of evidence can be for the variant interpretation workflows. So the functionally abnormal or the not variants get strong, and the functionally normal ones get strong, right? Because this assay is so good at separating. And this is the nice thing about BRCA1 is that there's enough variants in the database for us to actually get to that level. Okay, and then unfortunately, because there's not a lot of publicly available clinical data, we collaborated with Ambry Genetics, who have been spectacular collaborators. They gave us all the information they had for variants that overlapped our data set. And so that was 287 variants. A little less than half of them were variants of uncertain significance, or 110. And then Sean applied strong evidence for pathogenic, strong evidence for benign to any other data that they already had, and used the ACMG interpretation framework from 2000 that was laid out in 2015. And he was able to reduce the variance of uncertain significance by about 50%. So a few went from 5 or so went from VUS to likely pathogenic. But 49 went from VUS to benign, which is equally as important. And so that's 50%. And a lot of the genetic testing companies use kind of different algorithms in the ACMG guidelines. So this number could go up. He had also done this for published P53 data sets, where he was able to reclassify 63% of the variants of uncertain significance. Again, moving most of them to likely benign. And then P10, only 15% were reclassified. P10 is a weird case. P10 has basically no benign variation. So it's really hard to do that odds path validation where you have to, get them right in the benign side as well. And so that was really difficult. So he just followed ClinGen's variant curation expert panel for P10 and was able to reclassify about 15%. This isn't just us. There's a great MSH2 data set from the Kitzman lab at the University of Michigan, where they were able to move about 80% of their variants of uncertain significance, either to likely benign or just a few to likely pathogenic. And then these people didn't have a nice figure, but a study of DDX3X, which is a developmental delay gene or gene associated with developmental delay, they were able to reclassify 90% of the VUS in that gene as well. So this is happening, right? This is successful. We're able to actually use this data to reclassify VUS. Ambry Genetics in both of these cases actually gave updated classifications back to the ordering providers. So the patients actually got these updated variant classifications. So this is really awesome. Okay, so now we know that functional data can be pretty useful to reclassify variants of uncertain significance. So we want to scale this up, right? So this works, right? We should turn the crank. And so as part of a characterization center for the impact of genetic variation on function consortium from the NHGRI, I have a characterization center, or basically a production center, to generate more of this kind of data with Doug Fowler, who's another professor at the University of Washington. We call ourselves the Center for Actionable Variant Analysis because we try to look at or apply this to genes where that data can be immediately useful and impactful in the clinic. So we built a huge team. We've got everywhere from software developers to undergrads who are, my two undergrads are these amazing MIDI prep or Maxi prep machines. We've got two assays. So we use saturation genome editing, which is the assay I just showed you. We have another one for protein abundance. And it's a really simple assay. We just fuse the gene of interest to GFP. If it has a destabilizing variant, the GFP is dim. If it has a variant that doesn't cause any harm to the protein structure, it could be bright green, and we just separate those by FACS. And then sequence and count the variants in each FACS bin, right? It's pretty easy. Between these two assays, we think we have access to about half the proteome. So that's why we chose them. They're not, dead and green is not as informative as maybe you would like, but in terms of mechanism, but it works really well. So we spent a lot of year one of IGBF kind of optimizing these for production. We're able to really robustly get nice repetitive, nice robust data out of this with R-squareds in the 0.95 range for saturation genome editing, and we use PALB2. So PALB2 stands for partner and localizer of BRCA2. And it's like the third gene in line after BRCA1, 2, and then this would be BRCA3, we think. And so we have 10,000 variants in that and about 3,500 3-base deletions. And for the protein abundance assay, we built a lot of kind of standardized vectors where we'll be able to just drop genes in more easily so that we can run pilot experiments really quickly. And again, I'm bringing the slide back and just showing you for every about 120 bases that we query in the genome, we need a new guide and we need a new plasmid library, right? This is labor intensive stuff. My lab, what we pay at IDT is insane. So again, this is for 4,000 possible SNVs. Our experiments for the first year of IGVF are 10 times bigger than this. We're making 137 libraries, 137 guides. So I also run a COVID testing lab in my spare time. So we've been taking the automation that we built, that we have for that, and trying to apply it to a lot of these cloning steps. So this is a mantis. It actually looks like a mantis. It's very cute. It will dispense into 384 well plates. And this is like 96 well cloning. So this is 96 guides going in to their vectors, and we got 93 out of the 96 on this plate. It's awesome. So we're kind of working on automation for a lot of this. And again, our genes are chosen for clinical utility and for different use cases. We work a lot with the genes that are on the incidental findings list. So incidental findings is the special set of genes that are medically actionable, such that if they're found during either an exome or even a research study, they have to be returned to the patient or to the individual because medical management can ameliorate any of the problems that might occur from having that variant. And so a bunch of genes that we target are on that list, and those are labeled as ClinGen. Gregor is the new Mendelian disease consortiums from NHGRI. And these are genes that either give whole exome sequencing, whole exome people trouble, or they're potentially new disease genes, but they have missense variants. So, they're hard to predict whether or not they're damaged or not. And that's where a lot of these other genes come from. So we're super excited about SFPQ. Those are coming from families at UW, which is really neat. The other UW. CPIC, so these are genes that are important for pharmacogenomics. People who have damaging variants in these can be non-metabolizers for a lot of drugs and can be overdosed by things like warfarin and other drugs, not here, or they could be super fast metabolizers of drugs and aren't getting enough of a dose, right? So these are genes that fit those categories, CYP2C19 and G6PD. And again, there's a lot of hereditary breast and ovarian cancer on there because that is what I am personally interested in. So that's scaling new technology. So you might not have seen your favorite gene on some of those lists, and that is because we have no access to it. It's either not expressed or doesn't have the right complexes in the cell to really be able to measure its function. And so through another center, this is the Center for Multiplex Assessment of Phenotype. It's a center of excellence in genome sciences, also with Doug Fowler. That is what we're trying to get access to. So again, if you look up this incidental findings list, there's 73 genes. I think there's 79 now. They're pretty evenly split. About 28 of them are cancer genes, so like the tumor suppressors I was telling you about in the beginning. But the other half are cardiovascular genes, or genes that cause cardiovascular phenotypes. And many of them we just don't have access to, right? So they're not expressed. Those genes aren't expressed in that haploid cell line. And so we have really no way to get to them. So what we wanted to do as part of this grant is be able to take what we've been doing, what we first did in yeast, what we then moved into utilitarian cell lines into stem cells, so that we can get the variants into stem cells and then be able to differentiate them into a bunch of different cell lineages that may give us access to the genes or their protein products in the cell type where they are relevant. Okay, so the first gene we're going to look at is MYBPC3. 50% of hereditary hypertrophic cardiomyopathy is associated with MYBPC3 variants, and that is where the heart builds up too much heart. You can tell I'm the molecular biology person here. Anyway, but many treatments available treatments are available if you know you have a pathogenic variant in one of these in this gene and some others, and it can lead to heart failure if not monitored. And so MYBPC3 is a myosin binding protein. It sits between the actin filaments and myosin heavy chain, and acts kind of as a regulator or a brake, actually. If you, interestingly, if you look at the cells that are knocked out for MYBPC3 under the microscope, they kind of like almost beat themselves off the plate. It's kind of crazy. Anyway, so most HTM variants are non-sensor frameshift, but there's a lot of missense, and most of those actually are variants of uncertain significance. Okay, I'm going to show you the team. Again, it's Sean Fayer, who is an amazing graduate student. This is also always in collaboration with Doug Feller. And then Daniel Yang and his postdoc Clayton Friedman are the real cardiomyocyte specialists. Daniel is actually a cardiologist, and they kept us from doing really stupid things, which was great. So it turns out that cardiomyocytes basically shut off all genes except for the ones they really, really need. So the sarcomere proteins and things like that. So transgenes, you can't go in with a lentivirus, for example, it'll shut everything down. So what we realized we had to do was edit directly into the locus, which is hard. Nobody had really been able to get the number of variants that we would like to be able to get into these cells into IPS. And also they're human cells and they're diploid, which is super annoying. MYBPC3 cardiovascular risk is a dominant phenotype. So what we did is we went in and to one allele, and we knocked in a GFP and a pyrimycin resistance cassette. And this GFP comes on if you differentiate the iPSCs to cardiomyocytes. And the most useful thing here that we did is we deleted about 500 bases that we could then target in the other chromosome with a Cas9 to get, again, these last 6 exons of MYBPC3 in, with all possible single nucleotide variants in exon 32, plus a red fluorescent protein. So the cells that we want are going to be green and red, and then a blastocidin resistance cassette. So they should be blast resistant, pure resistant, green and red, if they're in the right place. Okay. And we had about 498 possible variants. We were able to get 494 of them into the cells in this first editing. Well, it wasn't the first one. It was after many optimizations, about 494 of them in, which is super exciting for us. So again, this is the reads from the plasmid library versus reads from the knock-in. This is all junk. And then this is the real stuff here up in this right-hand quadrant. And then we looked at this on a heat maps. This is every possible position in the amplicon that we sequenced. This is where the variable region starts and ends. And you can see that we've got some nice even coverage of all of the variants in that exon, which is really exciting. So what is the phenotype we're going to look at, though? We can get the variants in. What's the phenotype? So this is a little cartoon of cardiomyocytes expressed in the library. When variants, especially in the C-terminus of MYBPC3, are damaging, these variants don't get incorporated into the sarcomere, and MYBPC3 is degraded. So we figure we might be able to sort on low intensity MAPL versus high intensity, and build and calculate functional scores that way. So this is just a picture of kind of what that looks like. This is a cardiomyocyte. This is the wild type version on the one allele. You can see the nice stripes of the sarcomere. And this is a variant that seems to co-localize really nicely. Whereas this cardiomyocyte, you can see the wild type allele, but whatever variant this guy got, you can see that it's pretty dim you don't see any of this nice striated protein. And so this is like hot off the press data from like this morning. So I don't have any, I was in a rush, sorry. And so he took those cells and he sorted them into high and low bins of MAPL expression. And this is kind of, and then sequenced the variants that were in each of those bins and counted them and took like basically a weighted average of which bin the variant came from. And you can see this distribution. He really needs to rebin this, but you get a bunch right at both ends and then some in the middle. And when you look at just the variants that are in ClinVar, the blue ones are either likely pathogenic or likely benign or benign, and the red ones are likely pathogenic and pathogenic. And you can see those separate really nicely in this assay. So we're super excited about this. We would love other phenotypes, but this is what we're starting with. Okay, so we're able to edit these stem cells, which is really exciting for us. We're going to start going into other cell types pretty soon. And we've done a lot of work here, but we also need other people to continue. We keep telling the NIH we're going to do it all ourselves, but we really can't. And so we're trying to build some community to be able through two efforts I'm going to tell you about in a second. So the first is the Atlas of Variant Effects Alliance, and this started by like-minded people who wanted to kind of catalyze more work in this area. So again, Matt Hurls is a big driver of this, and he's really interested at the Sanger in rare disease that causes intellectual and developmental delay. And again, through a lot of his studies, he's finding a lot of these variants of uncertain significance in a lot of the families that he looks at. And so it was founded in 2020. We have 400 members from 20 countries. We host Mutational Scanning Symposium, which will be held in Hingston this year at the Sanger in July. So the registration just opened today. So if anybody is interested in that, please sign up. We also host the Variant Effects Seminar Series, or VES. Again, nobody needs another Zoom lecture in their life, but this has been great. It is organized by trainees, and it is PIs are not allowed to talk. So this is for trainees to talk about their research, which has been really nice. And again, the objectives here are to bring together data generators, consumers, and curators to really kind of talk about how, or figure out how to generate the data that the consumers want. And then by consumers, we mean like clinical genetics, the clinical genetics crowd. And then, obviously the end goal would be to produce an atlas of all, sequence function maps for, all clinically relevant genes. And the second community resource is MaveDB, which is a database for storing and sharing this kind of data. And, as these experiments have really taken off, there's about 3 million variant scores. Again, GFP has been done several times. Not all of them are terribly relevant to the clinic, but they're there, and having them all in one place is really important for a couple of reasons. One is people who are building the next generation of variant effect predictors, the computational crowd, they're there, they're curated for people to be able to download entire data sets, which is cool. The second thing is this is almost like the early days of microarrays or sequencing, where everybody was, the data was all over the place. It was being, some publications don't even publish their data, like the actual data set itself. And so this is kind of, almost in the protein coding field, really become the standard for where you have to put your data, which is super exciting. Okay. All right, so we have some community resource building, and then getting the functional data into the clinic, which has also been hard. So the first thing we want to do is we want to hook MaveDB up to ClinVar. So from talking to clinicians, this has been a very big learning experience for me, right? I'm a straight PhD. research person. And, we've had some great clinicians, very forward-thinking clinicians who have banged on our door and said, hey, you guys have all this really cool data. Where do I find it? And I say supplementary figure 2, and they say, no, no, that's not okay. And they're like, well, it has to get into ClinVar. We're like, well, how do we get into ClinVar? And how do we get it in, how do we wrap it up in a way such that it is usable to y'all? And we've been working again with the clinicians, and we're hoping to be able to hook MaveDB up to ClinVar to feed that data to them in a way that is digestible and immediately usable. We're not quite there yet, so it's a just started. And of course, working locally. So University of Washington has one of the oldest clinical genetics departments, in one of the largest, and all of their data from the last 20-30 years is locked up in random Excel files and and medical records, right? So how do we work locally with data that we have to cherry pick out of EMRs, and you really can't do that. So Andrew Shergachis has been working on unlocking all of that data, building what we call the Brotman-Beatty clinical variant database. And we're basically just paying people to pull all that data out of all of those electronic medical records and Excel files and get it into a database that's usable, so that if they have a variant of uncertain significance and we have functional data, we can get together in clinical conference, and which is a meeting they have every week to try to present cases. And we can get together and see if we can resolve that variant of uncertain significance. And we can also teach the clinical genetics community at our local institution how to use this data. And so we're trying to act both locally and globally to get this data into the clinic. All right, so I hope I've convinced you that we are heading down this road to ending variants of uncertain significance. But of course, there's lots of work ahead, and there's lots of people to thank. It clearly takes a village. I've mentioned a lot of this work is consortium based. So we work in a lot of teams, and this is part of our teams. And I'd like to, just so much work by so many people. And thank you all for listening, and I'd be very happy to take questions.
And I should say, or maybe you can say for the people online, they can ask questions in the chat.
I see a question in the chat. Somebody's asking if the RNA score is coming from single cell sequencing. No, that's too expensive. It's just bulk cDNA generation and sequencing of the variable region. That's all. And we can tell if a variant becomes depleted in the RNA versus the DNA, what that it is damaging to either splicing or expression somehow. Yeah.
You get community acceptance of moving VUSs into the benign category, because of course you just tested it one way in one setting, and maybe in other contexts, other backgrounds that are quite significant.
Yeah, yes, that is a very difficult problem. So that he's asking about moving variants to the benign category. So it's not the only evidence that's used, right? Functional data can never stand alone. So there's other evidence that it is benign, and whether that is actually allele frequency is very strong, but other variant effect predictors, which could be evolutionary based. Yeah, that is also what keeps me up at night, that it could be pathogenic or in some other category, but we're never going to measure every possible thing, right? And if you go back to that BRCA1 story where we had so many benign variants that were behaving in a way that said that the functional assay was measuring the correct thing, right? So for BRCA1, I think we're pretty sure that any other random variant would also behave in a benign, like the rest of the benign variants. But that does get harder when we move away from genes like BRCA1, P53, where those controls just really aren't there. But I mean, we have to do something, or those variants are going to be VUS forever.
Well, maybe it's fine for them to stay VUS until there's data suggesting that they are, in fact, pathogenic. And that the benign is a pipe dream that may not be achievable.
Maybe.
I had a question along the
Yeah.
same lines, and I wonder if it's actually harder to classify definitively the benigns, because if you have a narrow assay, don't mean narrow in a
Mhm.
negative way, but you
Right.
have to have, very
Right.
precise assays. The buses that do have an impact in what clinicians might call an in vitro setting,
Mhm.
it is an in vitro
It is,
setting.
yeah.
That still is probably, those are probably really important to understand. Whereas the benigns, you could imagine, maybe in this proliferation assay,
Mhm.
they don't have an effect, but.
Right, I mean, so you wonder, if it would be in the right context, if we're missing like a binding partner or something like that. In this case, this cell line happens to be exquisitely sensitive to double-strand breaks. So, and there's a lot of data out there and for BRCA1 and 2 that double-strand break repair is the tumor suppressor mechanism. So I'm like less concerned about that assay for BRCA1 than I would be for like, say that cardiomyopathy gene. But yeah, that's a real philosophical question, whether it's better to leave VUS as VUS or, take whatever evidence you can get and start pushing them toward the more definitive categories. So the ClinGen is switching to a points system. So there will be, VUS that are kind of pushing toward benign and VUS that are, kind of pushing toward pathogenic. So that might be, that might kind of answer your question there, where you're kind of pushing it toward benign, but not saying it's actually benign. But I think it's a philosophical, it's whether where you're comfortable, if you're comfortable leaving it more uncertain or not.
Are there other questions? Oh,
Mhm.
I was wondering, for classifying these, is there, I guess in these databases, how much do they care between maybe a dominant versus recessive phenotype? Because right in yours, you're not necessarily sure for the one.
Right. So we're really only looking, obviously, at loss of function because the other allele is not there. So we can do this assay in diploid cells to get that dominant negative, get it dominant negative effects. Sorry, what was the rest of your question? I kind of lost it there.
In these databases, are they classifying that type of information? Whether they are dominant or recessive, like, is that important for the pathogen?
Yes. So for most human genetics databases, we know, or I shouldn't say that. I know the hereditary breast and ovarian cancer ones, and they're all dominant because they're just the two hit, they're tumor suppressors. For other genes, whether it's known or not, whether they are dominant negative or loss of function, it's not always known. But I think if you were going to do an assay like this, that would be something you would you would try to find out before you did it, because obviously the dominant negative ones are not going to do anything interesting in a haploid line.
Yeah, I can ask it. Another question that I'm wondering, I don't know how much clinicians, and maybe it depends on what type of clinician, if somebody in a sequencing center, maybe they just want to know, is this a disease causing or not? But I'm curious how far you can get into the mechanism of different variants, especially if you use different assays.
Yeah.
Where you have some variants have an effect in one assay and not another, and can you actually get farther into predicting functions, mechanism for this variant?
Yes. So obviously because we've traded phenotypic complexity for scale in these assays, right? So again, live dead, green or not green with reporters, but we would eventually the library generation in these assays are kind of the most expensive thing. So we would love to be able to put them against, develop a panel of phenotypes and have a matrix of variant by phenotype. That's like the pipe dream of like a future state.
And what about you just proliferation assay, but under different conditions? Have you tried that? And how, I'm sure it's gonna be different for every.
We have to some extent. So we have taken, for PALB2, we're gonna have data at day 13, 17, and 21. So we'll be able to build like kinetics of the dropout as well. And we do see a set of missense variants that at day 13 look okay, but start to drift toward non-functional or toward the functionally abnormal state later on. And I mean, the thing is that the human data just isn't there to compare to. So we won't know kind of the biological meaning of that. And in terms of mechanism, yeah, so I think we learn a lot about the biophysics of these proteins from these mutational profiles. So I know like the Leonard lab in Spain, now he's back at the Sanger, is able to generate pretty darn good structures just from mutational data alone. Of course, they're doing a bit higher throughput. They're doing every possible amino acid change plus a lot of double mutants, which we just don't have the space for in our assays. But yeah, so I think we're going to learn a lot about protein folding. Editing assays, we're going to learn a lot about splicing. In just the VRCA1 story alone, we had exons that were like bomb-proof for splicing. Like none of the mutations affected splicing at all, and other exons where a full quarter, like a quarter of the single nucleotide variants disrupted splicing. Like, why are some exons fragile, I guess, and some not? And with an N of 13, the PALB2 is almost done. That'll give us an N of 49. That's still not enough to learn these things. So I think hopefully by the time IGVF is over, we'll have enough data to start learning some of those rules.
Any other questions? Yes. Maybe for the sake of time, we'll thank Lea again.
Thank you.
And then this is, and this also.